This first case study was in conjunction with Quotient and Janssen, and it's a model to think about how we bridge from a solution to a solid dosage form within a first-in-human study. So this was a first-in-human protocol where we had a SAD and a MAD, and a switchover from a solution to a solid dosage formulation within the first-in-human study. So the drug in question was poorly soluble, and in the preclinical species, there's unexpected differences between the species. And so Janssen really wanted to have the flexibility to look at different formulations and circumvent these risks within the first-in-human study, with the desire to still end up with a patient-friendly formulation at the end of the MAT. So if you look at the design here, we started the clinical study with a solution formulation in SAD period 1 and 2, and then switched over to look at SAD dose periods, to look at solid dosage forms in SAD dose period 3, and then continued with the best performing formulation into the remaining periods as well. So this really allows us to start your clinical trial very speedily with a simpler formulation, but in parallel, develop a patient-friendly and scalable formulation for phase 2. And all of this was done within a 10-month period as well. This is a second great example of how we've helped a customer to move from first-in-human to proof of concept. This is for MUI, a US-based biotech, which were taking an oncology molecule with poor solubility into a first-in-human study. And because the drug was developed for an oncology indication, you would think the most common route would be to go straight into patients. For this particular molecule, the decision was decided to go into humans because it was determined by the need to rapidly identify an enabling formulation and a dose, which then would enable a swifter advancement into the target patient population. We looked at multiple enabling formulations in order to accelerate that transition. The formulations were selected based upon the physicochemical characteristics of the drug substance. And we looked at a blending capsule with surfactants, a lipidic formulation, and a spray-dried dispersion as well. This was a SAD study using an adaptive design available to enable testing of the performance of different formulations and having the ability to really precisely adjust the dose in response to the safety PK data that was emerging, and still allows to select the optimal formulation for phase 1B and 2. The PK modeling was also used based on in vitro and preclinical data to predict doses first in human and to follow on for phase 1B trials, which were implemented in trials. And Quotient were able to also then scale up that formulation seamlessly to supply product for future efficacy trials. All of this was completed seamlessly within 12 months. So through those integration strategies that I've described and using an on-demand manufacturing strategy, we can integrate product manufacturing clinical testing in the early development program. So a development team can have significant time savings. Fundamentally, the first-in-human can start with a simple formulation using minimal stability data and transition into an alternative drug product or screening different technologies within the SAD portion of the study. And in addition, the drug product manufacturing know-how can seamlessly transition into POC trials. And in summary, we can expedite to an average of 11 months from a first-in-human formulation development through to the availability of clinical supplies, with the time saving of 15 months, all of which facilitates drug development for molecules on the accelerated submission pathway and de-risks the CMC.